Good morning, everyone. This is a talk that I've done with two fantastic collaborators, which I'd like to acknowledge here. First of all, this is Alex Boren on the left, who is a third-year environmental science student at Berkeley, and Ali Byrne, who is a postdoc in the same department. We met actually 2 years ago at a conference on amphibian disease, which really speaks to the importance of those conferences, and I would like to applaud here the work done by Christina Davy and all her team at Carleton to put a fantastic CHS so far. And so the conversation we had 2 years ago was about disease and how it was very interesting to see that the pathogen that you may have heard about, Batrachochitridium salamandrivorans, is very much present in Europe and killing a lot of salamanders, but it's not in the North America. So this talk is also about presenting the potential of an invasions and how research is shaping in the preparation to that invasion. So why now? Clearly, it has been some time since the pathogens was discovered in Belgium 10 years ago. And what we would like to do through this talk is really pause, reflect, and reassess the finding of these 10 years of research. There is a lot of data that we've gathered, a lot of papers that have been published. So what does it tell us, and how do we reflect on what has done so far? So as I mentioned, there's several trends that could be observed, and here I'm going to focus mostly on the methodological trends, what has been done, how, for what purpose, but also geographical trends, as I alluded in my first slide, the dichotomy between the European context where the pathogen is present and the North American context where it's absent, and we looked at those differences. So how did we do it? We used two search engines, the one at Berkeley and the one on Google Scholar, and through saturation of the 200 results showing a lot of congruency, we extracted 73 papers that we're going to use in this study. So by any means, it's not intended to be a perfectly exhaustive list of all the Bsal literature, but it's instead working as a large enough data set to eliminate some of the relevant trends and illustrate their impacts here. So what made a paper relevant to our study? It had to be a laboratory experiment pertinent to Bsal and use living animals. It could also be a report of a dead animals found in the field or in captive collection where the animal died from that pathogens. But also we are looking at swabbing in the field, how monitoring for the disease is done, and what papers have shown us about this swabbing. So first, a quick overview of 10 years of Bsal research. Those 73 papers are scattered through time. There's an increase all the way to 2020 with 13 papers in that year, and then you see a decline. It's certainly not that the community is losing interest on Bsal, but I think it reflects more the COVID period where a lot of campuses were closed, therefore experiment didn't go on. Going in the field was also very complex and required a lot of authorization when it was even possible. So I think there's a lot of data that are dormant at the moment and will probably pick up by the end of this year and next year as people sort of analyze all their data and publish their findings. The first thing we're going to turn our eyes on is the methodological trends. So what people have looked in research. So clearly there is half of the papers that are about swabbing, and then another third about experiment. The others are about deaths in the field. What's interesting to see here is that those different type of papers are reflecting what we saw in the general trends, with a peak between 2017 and 2021, mostly in the experimental part and also the swabbing part of those papers across the world. Now, when we turn our eyes to just the death of animals due to Bsal, and we are over 4,500 animals that have died, that's where the striking results is in our study, is that more than 90% of those animals died through experiment. They didn't die in the field through infection, but they were actually used in the experiment to test some questions about Bsal. And then there's about equal amount that died in the field or died in sort of pet store, the pet trade or museum. The cause of the death, if you look, also we were interested to know what species have been dying, and the red here is the experimental death with 1,000 of our newts here in North America, followed by 300 of the fire salamander in Europe. Clearly, those species are not endangered. However, if you look down the line, there are several species that have IUCN status, either near threatened, vulnerable in blue, endangered, or even critically endangered. So it begs the question about the use of these animals into experiments, because in all cases, these are not animals that died in the field, but clearly animals that were used in the experiment to test their vulnerability to the pathogens. And if I only took a snapshot of those species just to put it on a graph, but as we noted, there's 84 species that have been used for Bsal. Looking again at different trends here, this time we looked at what species have been used. Again, we have the two top species here, and the highest number again is for experiment, and then some species at the end have used also in the field. What's interesting on that slide is I added the little symbols of a salamanders to denote where the species are not exposed, not naturally exposed to Bsal, because it's not there. So as you know, the North America doesn't have, therefore Bsal, therefore the newts is not possibly exposed naturally in the field, despite being the most used in the experiment. And that's also true to our North American anurans, such as Cattleya, pipiens, on the middle of the graph here, that have been tested, yet they are not possibly exposed yet to the pathogens. So to wrap up with the methods, we are looking at the primary purpose of these experiments themselves. That leads to a lot of the deaths here, 4,200 are within experiments. And half of it is to know the pathogens, whether we're looking at papers or the number of deaths. Almost 50% of them are to understand the pathogens. And then there is half of the deaths are to understand the host response, and then there's a small number, about 10%, which is about mitigations, where you could think that after 10 years, those numbers would ramp up as we understand better the pathogens. We also have a lot of knowledge of its cousin, BD, so I would have anticipated a lot more papers about treatment to justify the use of animals in the lab, as opposed to simple understanding the pathogens. Now, what about geography? As I mentioned in my starting, big differences across the Atlantic. On the west side, we have North America, where the pathogen is not present, and in the European side, it's present across a lot of countries. So, where is Bsal research being conducted? The leader here is USA, again with no presence of Bsal, and then we have the countries where it's found the most in Europe: Belgium, Germany, Spain, and Netherlands. So again, a very different picture across the pond. Lots of countries that do have the pathogens are doing a combination of experiments and swabs, while when you look in North America, there's a lot more papers that are experimental based as opposed to the monitoring to prevent the entry, or at least to alert a possible entry of the pathogens on the continent. How did animals were dying? Again, in the USA, a lot of these animals were dying due to experiments, no field deaths, obviously, and also no presence in the museum. In France, same thing, a lot of experiments. It's not been noticed in France yet, and in Belgium, these are experiments. The number in bracket is the number of papers. You'll notice that Bsal has been recorded in the UK, but only in the pet trade and not in the not in nature. When I do a country comparison, and for the last couple of slides, I'm going to focus really on Belgium and the USA, which are sort of at the top of the previous charts, and that's where, again, where the difference is striking. Whereas in the USA, Wild-caught animals represent 71% of the animals that have been used in the experimental design versus 28 that were captively raised. But in Belgium, we have almost the opposite, with close to 80% of animals were raised for the purpose of experiment, and only 20% of them were caught in the wild. I sort of wonder why this is. I don't know exactly about the differences in animal care protocols and ethics that are going on in these two countries when it comes to approved experiments, but that might explain some of the differences we notice here. Going a bit more in details to finish, here's the difference in terms of the life history stage. A lot of adults in North America, almost 70% of the animals were adults, while in Belgium we have a more evenly split between adult and juveniles, and a lot of that were not specified in the papers. So again, looking at the type of species, we have the newts, which is prevalently used in the USA versus the salamander in Belgium. Does it explain the differences? Is it easier to capture larvae or experiment on larvae in Belgium than it is in the USA? Why is there such a focus on adult here? I'm not sure. I don't have the answer at the moment for this question, something that probably needs to be further investigated. And finally, in terms of the experimental source of the animals, here there's not much difference. There's a lot of animals that are non-threatened in the USA and in Belgium, but there is still a significant number of animals in the USA that belongs to one of the 4 threatened, vulnerable, endangered, or critically endangered status from the IUCN, where that number is very minimal. In Belgium, it's only 24 animals that have been used, so like 10 times less than it's been in the USA when you look only at vulnerable, endangered, and critically endangered. Again, that may reflect also the way the authorization to do these experiments with animals are approved in the 2 different countries. So to finish, I think my last table is really to look at those 2 species that have been used. The red-effed newt in the USA and versus the fire salamanders in Belgium. As I recall, for you, the pathogen is present in Belgium, but it's not in the USA. The number of swabs itself is very different. It's almost a 10-fold difference between the number of swabs that are done in the USA, which I think is a good thing. It's really a preventative approach that's done in terms of swabbing across the country when USA is trying to avoid or really pinpoint where the pathogen could potentially arrive on the continent and when. Now the difference that I've also highlighted before is the number of experimental deaths between the newt and the salamanders. A lot more newts have been used to understand Bsal in the USA that have been done in Belgium, with 1,000 dead newt versus 236 newts, salamanders, in Belgium. I've already mentioned the differences in life history stage and also where they come from. 86 animals were wild caught for the salamanders versus the entirely 995 animal newts were captured in the wild for the newt. So moving forward, I think it was time to do this work to really reflect on how we study disease, amphibian disease in particular, and look back at what has been discovered. We didn't look at the particular discoveries and knowledge, but more about the use of animals in the wild. So I think it's time for us to reflect on how we use experimentation versus field swabbing, what type of demographics we are studying and the impact it could have on potential population where we capture those animals, as well as the geographic distribution of the research. I think as technology improves and our understanding of techniques improve, it's also about time that we use maybe different approach, like genomics, for instance, sense to study those diseases and those pathogens and minimize animal loss in general. And with that, I'd like to thank everyone for their attention, and I will take some questions.
Okay, so habitat loss is a problem for a lot of species, but amphibians have been pretty hard hit. It's been pretty severe and pretty widespread. Southern Ontario gives us a pretty extreme example because we've lost about 72% of our historic wetland coverage in Southern Ontario. Pelee Island, which is located in Southern Ontario, is actually located in Essex County, where these losses have been most severe, but it's home to one of my favorite animals, this smallmouth salamander. So their populations have declined as a result of habitat loss. So Pelee Island itself was historically covered in a giant wetland, which was largely drained for conversion to agriculture in the late 1800s. And then now there's less than 6% wetland coverage on the island. So we've been studying this population since 2015. And one of the things that we do is we survey the potential breeding sites on the island. So there's about 30 or so that we try and track every year if we can. We look for, we use dip netting survey. We identify the presence, but also the relative abundance through catch per unit effort. And we're monitoring both constructed and natural ponds when we're doing that. We're also collecting a bunch of habitat data, things like the canopy cover around the pond, the water temperature, leaf litter in the substrate, the presence of crayfish burrows, a whole bunch of other variables that might be relevant to the quality of those habitats. And the reason we're doing that is to try and get a sense of what makes one pond better than another, right? And so what makes a pond good? We tried to address this in a paper that we published in Wetlands in 2020. So some of the things that we found were important were high amounts of canopy cover around the pond and the presence of crayfish burrows. Those were associated with the presence of larvae in those ponds. And larval density, the density of those salamander larvae increases with the amount of leaf litter in the substrate and also the presence of submergent vegetation. Just these are key indicators of habitat quality. And the other thing that we took away from that work was that these constructed ponds that are existing on Pelee that have been built to help amphibians are not currently suitable habitat. They're not good habitat. So only about a third of those constructed ponds that we surveyed actually had any larvae in them. And the ones that did had 4 to 10 times lower catch per unit effort than compared to a natural pond. And so the question then becomes why? What's explaining that variation? And one of the things that I think plays a big role is just the pond age. So it takes these ponds a really long time to reach a naturalized state where they become highly suitable wetlands for the salamanders to breed in. So here's a graph that shows sort of pond age on the one axis and pond quality, which is our composite variable, our indicator of pond quality. And because we've been studying these constructed ponds, we can actually use the known age of the ponds to sort of give ourselves an estimate of the naturalization period, how long it actually takes these ponds to become suitable habitat. And we can look for how long it takes them to reach that threshold of the observed range for suitable or high quality natural sites. And the takeaway is that it takes probably at least 20 years for these ponds to reach a period where they're actually comparable to the natural pond. So that has implications for whether we can use biodiversity offsetting as a reasonable strategy or how we actually incorporate this as a conservation measure. But the other thing that we want to think about is how might we shorten this period? Can we get this to be less than 20 years? And if so, how do we do that? So let's go back to this pond quality index. We created a variable using what's an analysis called principal components analysis, which takes all of our habitat data and collapses it into a single variable of that we can sort of think about as pond quality index. And we can look at the habitat variables that actually go into creating that pond quality index, and those are the ones that we should think about when we're trying to identify how to improve these ponds, how to manage them, or how to construct them better. So these are the variables here. The things that distinguish between a poor quality constructed pond and a high quality natural pond are things like pond water temperature, the amount of canopy cover, so higher canopy cover is better, the larger surface area, the presence of submergent vegetation is better, the absence of fish is absolutely essential, the amount of leaf litter in the substrate. So we can actually use these variables then as the things that we want to target when we're designing or assisting these ponds through that naturalization period. Okay, so just as a sort of a connection to this story, the Nature Conservancy of Canada in 2020 actually constructed 3 ponds with the purpose of helping salamanders on Pelee Island. And because we had already been actively collecting this data, we were in a position to provide them some ideas about the design and how to and where to place these ponds, how to make them and where to place them. And so, yeah, we were able to actually give them some guidelines. One of the key things that we suggested was making sure they're tight against those tree lines, and then supporting the naturalization process by planting trees around the tree line. So these are things that the NCC actually did. And that will not only help with the leaf litter deposition into the pond and help the naturalization process, but also help to support the growth of that canopy cover, which is in itself also important for managing water temperatures in the hydro period. Now, 2 of these ponds actually dried up in the first year when they were after they were constructed, but we were able to identify that as a problem, get them deepened, and then in the second and third years, they actually held water for a long enough hydro period that they were a good salamander habitat. So we have these new constructed ponds that were constructed using an evidence-based approach as opposed to a haphazard-based approach, and we want to try and get a sense of whether or not our evidence-based approach is maybe shortening this naturalization period. So what we can do is we can statistically remove the effect of pond age and look at the residual pond quality index. So these values on this graph show you the pond quality index after you've controlled for the age. And what we see here is that these 3 ponds that were constructed by the NCC following our evidence-based sort of design actually have a higher quality than you would expect or predict based solely on their age. So we are looking like we're shortening that naturalization period. Another pond up here is similarly performing well. This is another different constructed pond. It was constructed much earlier in 2003 by a different group, but it's also high quality and punching above its weight compared to what we would expect based solely on its age. So I think together what we can say is we can probably speed up this naturalization process if we're strategic about how we build them in the first place, where we build them, and how we help them along. So the question then becomes, well, how or are the salamanders actually going to use these ponds once we create them? And so we've used a drift fence pitfall setup to try and evaluate that. So these constructed ponds, as well as some comparable natural sites, we set up pitfall drift fence setups around those ponds to catch the emerging metamorphs. So when they're leaving those ponds, to catch any metamorphs that emerge. And so if you've never seen one of these, what happens is the emerging salamander comes out of the pond, hits the fence, walks along the fence, and then plunks down into our trap, where they're safely protected from raccoons by these flat rocks we have to put over them. So one of the exciting things is that, in fact, all 3 of these ponds are now being used by salamanders. So the first pond was used in the first year. The other 2 ponds, after they were deepened, they've been used for 2 consecutive years as well. So all 3 of these ponds are actively being used already by salamanders, and they're also producing metamorphs. So they're contributing in a demographic way as well. This salamander actually is the first salamander to ever emerge from those constructed ponds. It's kind of cool to have this sort of tangible piece of evidence-based conservation outcome right in your hand as manifest as a real animal. It's really exciting to see that. Another thing that we need to think about, though, in the context of salamanders when they're emerging is where do they go when they come out of these ponds? And so the salamanders in these ponds, we have an array of our pitfall traps. That's these white numbered circles. And then the arrows show how many salamanders we captured in any of these traps. And what we're seeing is that the salamanders aren't necessarily going towards the forest where we would want them to go. They're kind of just going in a scatter shot in every direction, and probably a lot of them are wandering off towards the field area where it's really inhospitable habitat. They're going to get picked off by a predator, and there's no suitable cover for them to hide. So that just reiterates the importance of adding and supporting these pond restoration projects by providing a large amount of coarse woody debris or other places where these salamanders can hide to safeguard them against desiccation, which is pretty extreme in July and August when these animals are emerging from the ponds, but also from predation. I should note also that every day that we were checking these traps, there were new predator footprints in and around the pond, either raccoons, wild turkeys, or herons. So there's a huge amount of predation pressure on these animals when they're trying to get out and start their new life as a terrestrial salamander. Now, in the first year of this project, we didn't have any of these metamorphs actually being smallmouth salamanders. They were all unisexuals, which was a little bit disappointing, but perhaps not that surprising. But in the second year, we actually had a full third of the salamanders that emerged from that pond as juveniles were actually smallmouth salamanders, which is really encouraging because that number is much higher than we're seeing at other sites on the island. Again, suggesting that if we build it, they will come, and potentially we can actually use this model to try and develop high quality habitat for these animals looking forward. Again, it looks like in this picture, based on our data from 2022, that maybe the salamanders got the memo that the forest is that way. It's hard to know whether that's a tangible sort of result that's going to continue, but I'd like to think that maybe over time, as the pond naturalizes, that they'll start moving in that direction. But I think even still, we just need to make sure that we're supporting the margins of that pond with forest cover and other cover objects like coarse woody debris or rocks. Okay. So let's just summarize, like, what can we do to create these salamander ponds better? So a lot of these are pretty straightforward. First thing to do is make sure those ponds are built within 1 kilometer of a source population. That ensures that they can actually naturally be colonized and maintain some connectivity over time, over the longer duration. We also want to, again, support the canopy cover formation by planting trees and also positioning those ponds close to the tree line. So that supports not only the formation of a canopy cover, but also the deposition of leaf litter. So again, placing those ponds immediately adjacent to existing tree lines, planting trees around the margin are all really, really important aspects of increasing that pond quality and reducing that naturalization period. You also need to make sure they're big enough and deep enough to be demographically valuable. Really small ponds might be used, but they're not contributing anything over the long term in terms of the population and supporting our conservation efforts. So we need to make sure they're big enough and deep enough to be demographically valuable. On Pelee, what we've seen is basically that means they have to be at least 200 meters squared in surface area or larger. And as they get larger, they get better. So bigger ponds are better. They need to hold water for at least 2.5 months of 75 days in order for salamanders to reach that metamorphosis stage. So you have to dig them deep enough that they hold water that long. How deep is that? Well, you just have to kind of see, and that's kind of what we saw happening with the NCC on the island. They dug it, and then they had to go back and dig it a little bit deeper next time. But you need to be careful that you don't dig them too deep, because we've also seen a lot of the constructed ponds on Pelee have been dug way too deep, and they are ultimately all colonized by fish. And once the fish get in there, they're permanently unsuitable for the salamanders until those ponds eventually fully dry up and the fish are extirpated from that pond. But in practice, that doesn't happen very often if in those really deep sites. And so we just need to make sure that they're not too deep. That's actually even worse than digging them too shallow, which you can go back and maybe dig them a little bit deeper next time. So ponds need to dry up completely, at least periodically, in order to maintain some level of habitat suitability. And then the final thing that we need to do is we need to support the wetland vegetation, the formation of that vegetation using things like wetland plugs, and also, again, supporting it with cover around the pond margins. Okay, so I just want to acknowledge a number of the partners. We also received funding from the Species at Risk Stewardship Program from the province of Ontario, and Scales Nature Park has been a big collaborator on this project as well. So thanks, everyone.
Hi, everyone. Thank you for being here. So my name is Anaïs, and I'm finishing a master's degree at Laval University in Quebec City. And today I'm going to talk about the integration of data from different sources to evaluate impacts of forest management on spring salamander populations. So first, a little bit of context. So in forest ecosystems, the main anthropogenic disturbances of wildlife habitats are due to logging operations. So indeed, wildlife can be affected by canopy loss, which induces humidity levels to decrease and temperature to increase. But it might also be affected by road infrastructures, which are required to manage timber harvesting and also wood transportation. And this might be a source of sedimentation into watercourses. So all these changes might reduce habitat quality and have a potential impact on amphibian populations. So what is of interest in this project is that this might be particularly an issue for spring salamander populations. They inhabit headwater stream in eastern North America. And the species is widely studied to evaluate impact of anthropogenic activities. And the reason is that the species is particularly sensitive to forest cutting history and fine sediment inputs. This might be particularly due to their long aquatic larval stage, which could last up to 6 years. And meanwhile, the individuals are less resilient. And also the project is of conservation concern since the species has been designated as threatened in Quebec since 2011. So the objective of the project is to quantify the impact of forest operation on stream habitat by looking at the repercussion on spring salamander populations. So to do that, we use data set from different sources. And the aim in the long run is to evaluate current measures of protection for spring salamander habitat. So we based the study on 3 hypotheses. So we expected the abundance of spring salamanders to increase with habitat quality, with the main parameter being deciduous forest adjacent to the stream. We also expected the abundance of individuals to decrease with high cut surfaces and also to decrease with high density of road infrastructures for the reasons I introduced before. So now for the method, so the study area is located at the extreme southeastern Quebec, and this region represents the northern limit of the species distribution, which is associated to the Appalachian Mountains. So we can see here all the occurrences of the species we were able to gather in Quebec. So to respond to our objective adequately, we had to separate all the data we were able to gather in 3 spatial scale of analysis. So first we have the fine scale, which represents the survey of microhabitat in streams. So the data we collected for the project in the summer 2022. Then we have an intermediate scale where we combined professional surveys with habitat characteristics that were carried out between 2011 and 2022. And finally, we have the broad scale where we integrated data from professional surveys and citizen science, where we kept the same temporal scale as the second scale. So first, for the fine scale, we sampled 61 stream sections of 50 meters, and the sampling consisted of counts of individuals by the method of active stream searching. And we also collected water samples to be able to extract environmental DNA to detect the species in the stream. And with all that, we collected habitat characteristics. So with this data set, we analyzed the data with an Bayesian integrated model. So to build our model, we chose variable based on our 3 hypotheses. So for the habitat quality, we have the deciduous forest cover, but also other characteristics that we thought might have an influence on our population, like the density of potential flow beds, the dominant substrate in the stream bed, or also the average stream width. For the second hypothesis, we have the cumulated cut surface areas for the past 10 years before the sampling. And finally, for road infrastructures, we have the density of forest roads and the distance from the nearest upstream culvert. So for this first scale, the results are that deciduous forests cover best explains salamander data, and we did not see any effect of the other variables that we put in our model. So you can see here that the prediction shows that we might find higher abundance of spring salamander when the adjacent forest is deciduous, and it's most unlikely to find any individual when the forest is not strictly deciduous. Then we have the intermediate scale. So here we combined data from 7 professional surveys, which represented 519 stream sections. So here the data consisted of counts by active search, and they also collected common habitat characteristic. And that's all I can say for this part, since the analysis is still in progress. So I'm going to move on directly to the last scale, which is the broad scale. Here we integrated data from 13 professional survey and also citizen science data. So to be able to integrate the two types of data, we had to change the experimental unit. So here we ended up with 1,432 cells of 1 square kilometer. Here the data were detection of the species for each cell of 1 square kilometer. And we also used a Bayesian integrated model to analyze the data. So we follow the same process as the first analysis to build our model. So for the habitat quality, now we only have data that we were able to extract from georeference data. We did not have any variables from the field. So we ended up with density of deciduous forest cover and density of potential flow beds. For the second hypothesis, we still have cumulative cut surface area. And for road infrastructure, this time we only have the density of forest roads. So the results here are the same as the first analysis. Deciduous forest cover best explains salamander data, and we still have no effect of the other variables. And we can see that the prediction shows that the probability of occupancy of the species increases with the density of deciduous forest cover. So if we go back to our hypothesis, we might say that with the two analyses we finished, we have validated our first hypothesis, which is that the abundance of spring salamander increases with habitat quality, especially with deciduous forest nearby the streams. Yet we did not see any effects of the other variable we took to evaluate habitat quality, which might mean that the streams that were sampled showed the right characteristic of habitat of the spring salamander habitat. For the second hypothesis, we did not see any effect of high cut surface area, and also no effect of high density of forest roads, nor the proximity of an upstream culvert. So does logging activities and infrastructures affect spring salamander population in Quebec? We might say with our result that we have no evidence for a 10-year period, and also that this might mean that the current measures of protection for spring salamander habitat seems to be sufficient. If we want to go further, there are other variables that could have an effect, but we could not evaluate them because of data availability. But it could be interesting in future research to look at the short effects like recent constriction or recent upbursting nearby the stream. So there can be different impacts between the cutting treatments. And also another thing that could be really relevant are the condition of roads and culverts, if they've been restored or if it needs to be restored, or if it's been well installed, all that kind of thing. So that's all for me. Thank you for your attention.
I'm a master's student at the University of Ottawa in Dr. Julie Leo's lab, and I'm going to be talking to you today about using species distribution models from conservation for long-tailed salamanders.
Can you not turn your head? Speak.
Yes.
Directly.
Okay. Sorry. And I was a collaborator on this project. Kagan Finn was the lead author, and this was part of his master's thesis. So. Before we start, I do want to acknowledge that our independent survey data was collected on Treaty 7, which is the ancestral territory of the Blackfoot people. So translocations are an important conservation strategy to reduce the loss of biodiversity, and they're expected to be used more and more frequently. However, of the current translocation studies, only about 50% of them are considered to be successful. And so there's a need for tools to be able to increase the success of these translocations because our conservation resources are often limited and they're costly. And one way we can do this is by using species distribution models. So to generate a species distribution model, we use coordinates of locations of where a species has been found across the landscape. We extract information from GIS data sets about the environmental conditions that underlie these these presences. And then with this information, we generate a model that differentiates between these conditions and either background conditions or conditions where the species is known to be absent. Then after these models are reprojected back across space to illustrate places where the probability of occurrence is high, which can also be interpreted as suitable habitat. And there is evidence that these models can be useful to increase the success of translocation. So in 2020, Bellis and colleagues did a study where they generated species distribution models for ectotherm species that were included in previous translocation studies. And they determined the habitat suitability of the release sites for these translocations. And they found that the habitat suitability of the release site actually explained the most variation as to whether these translocations were considered to be a success or a failure. And so this indicates that these models can be useful to increase our success of translocation studies. However, species distribution models don't always make accurate predictions. So there's a need to validate these models and to test our model settings, because what's best for one species might not be best for another. So for this study, we focused on the long-toed salamanders. They're a small pond breeding amphibian, widely distributed in Western North America. And although they're pretty common throughout much of their range, at the eastern range limit in southwestern Alberta, represented by the star on the map there, they actually are a species of special concern. And within the province, they've been extirpated from many high elevation portions of their range. And so this has motivated the consideration of conservation translocations to reintroduce these salamanders back into their historically occupied habitat. There we go. And so this map here is our region of interest in southwestern Alberta. Those boundaries represent 3 protected areas that we are looking at, and they are Castle Provincial Park, Castle Wildland Provincial Park, as well as Waterton Lakes National Park. And we identified 36 potential release sites within these areas, and they're all in high elevation, fish-free, and they currently do not have any known populations of long-toed salamanders at them. So our main goal of our study was to prioritize these potential release sites for translocation. And we did this by developing useful species distribution models, by testing model settings and accuracy. And then we also wanted to explore the relative ranks of these release sites under current climate conditions, as well as future climate conditions. And so we tested 3 key modeling decisions to generate these models. The first was the type of environmental variables included in generating them. And so we had climate only models and then climate plus models, which had climate and non-climate variables. And then we tested the type of background points used. So we had randomly generated background points, and we also used target group background points. And then we also tested alternative study extents. We had a range-wide model, an ecoregion model, and then a genetic one. The genetic boundary represented the eastern subspecies, and we also had a political boundary. And so I'm not going to go into the detail of these just for the sake of time, but I'm always happy to discuss them afterwards. And altogether, we had 16 models generated representing all of these different combinations. So next, we needed an independent data set to be able to validate these models. So Kagan went out in the summer of 2022 and he sampled 110 sites, and he got 57 presences and 53 absences to get in our region of interest. And so I'll just jump right into the results. The first result that we saw is that our modeling decisions impacted our model outputs. So this figure here shows the 16 prediction surfaces from all of the models that were generated. The warm colors are areas of higher predicted habitat suitability, and the grayed out areas are considered unsuitable habitat by that model. And then the percentages next to each surface are the amount of area that was predicted to be suitable by that model. And as you can see here, changing the decisions had a substantial impact on the amount as well as the distribution of area that was considered to be suitable. Additionally, I want to point out that some modeling decisions had larger impacts than others. So if we look at those 4 top models generated with just the range-wide extent, we can see that when changing the different types of background points used to generate the models, there was a substantial change between 40 to 50% of the amount of area predicted to be suitable. However, when just changing the environmental variables within each background type, we saw little to no effect here. So our second result is that not all of our models passed independent evaluation. So this table just shows our models generated with all their different combinations of modeling decisions. And the last column there is the metric that we use to determine model accuracy. And we used AUC, which is the area under the receiver operating curve, and a threshold of 0.7 tends to determine if a model is accurate or not. And so of our 16 models that we generated, only 7 of them had AUC values greater than 0.7. And so going forward, these are our passable models that we had. So just to recap with the modeling decisions, they had substantial impacts on the predictions as well as the accuracy of our models. And then it's also, we can infer that it's important to run these sensitivity tests to determine your modeling decisions. But now that we have our top performing models, we want to use them to prioritize these release sites and to be able to make recommendations for translocations. So our third result that we saw is that the ranking of release sites also varied between these different models. And the table here shows just 5 out of the 36 sites that we previously identified. And the rank of each site as determined by the predicted suitability score of that model is shown in the columns. And so this is a really important result because it shows that depending on the decisions that you're using to generate your models, it can have a really big impact on which sites are actually determined to be the most suitable for those species. And in some cases, it was really drastic. So if you look at the third row down for the Lost Lake, if you use the 4th model, this was the #1 prioritized site. However, if you're using the 5th model, now it's number 18 out of 36, and we wouldn't really consider that to be one of our best sites. So that was pretty interesting to see. So we saw that we have a lot of variation in how these models are ranking sites. So in order to make decisions, we took an average ranking and ordered the sites based on this average rank. So this map here shows our 7 models in our region of interest, the 7 models that passed independent evaluation, and they were summed together. So the dark purple colors represent areas where more models agreed that that area was considered to be suitable. And then the average ranking of the sites are the values, and our top 10 release sites are represented by the red circles. And so we can see that these top 10 sites are pretty well distributed throughout our 3 protected areas. So that's really good to see that they're not clustered in one area. But this is under current climate conditions. We wanted to see how this would look when we're looking at future climate predictions. So we projected our models under a moderate climate change scenario as well as an extreme climate change scenario. And then we wanted to see if which sites would remain suitable. So when just looking at the extreme climate change scenario and then also accounting for places where our model was forced to extrapolate its predictions, only 3 of our top 10 sites remained to be predicted as suitable. So this was also really interesting to see. So just to recap overall, we found that the key modeling decisions not only had a large impact on the model predictions and accuracy, but it also influenced how our downstream recommendations are going to be determined for this conservation action. And finally, it's really important to explore the suitability of potential release sites under current climate conditions, but also it's important to test how future climate predictions are going to impact these release sites because we want our populations to be able to be supported in the future as well. So thanks for watching. I just want to acknowledge Kagan Finn again because he was the lead author on this study, and then also our supervisor, Dr. Julie Leah, and everyone who funded this project.
Awesome. Yeah, so like he said, my name is Justine, and I'm excited to talk to you all about Jefferson salamanders and road mortality. Okay, so unsurprising to many of us here, roads are negatively impacting wildlife worldwide. Roads contribute to habitat loss, fragmentation, and mortality. And animals such as pond-breeding amphibians, like wolf salamanders, are quite susceptible to road mortality. They have to migrate between 2 types of habitats, so forest and wetlands, to breed and to overwinter. They're non-reactive to incoming vehicles, and they're small and hard to avoid on the roads. So the target species of my research is the Jefferson salamander and its unisexual dependent, which make up the Jefferson salamander complex. Going forward throughout this presentation, anytime I refer to the Jefferson salamander, I am referring to the Jefferson salamander complex. I'm not going to delve into any of the wild world of Ambystoma genetics today. If you have any questions after, we can maybe talk. So Jeffersons range from Vermont to Illinois and Virginia to Southern Ontario, and both species require mixed deciduous upland forest and ephemeral wetlands to complete their life cycle. Both the Jefferson and the unisexual are considered endangered. There are only 21 extant populations left in Ontario, with unisexuals and Jeffersons coexisting in every population in Ontario. And one of the biggest causes of their endangerment in Canada is from roads. So here's a little infographic of the life cycle of the Jefferson salamander. In the wintertime, they overwinter in mammal burrows below the frost line. And in the early spring, when the pond edges are just starting to melt, they migrate en masse to the breeding ponds. And then in the late spring, once they lay their eggs, they leave the breeding pond and spend the summer foraging in the forest. In the late summer and early fall, the newly metamorphed juveniles disperse from the breeding pond. So if a road were to bisect any of these habitats, like what's happening in this infographic, it would make them susceptible to road mortality when they have to migrate between the forest and their wetland. So there is a population of Jefferson salamanders at a site in the Greater Toronto area. The exact location of the study site is withheld because of the endangered status of the species. The Toronto and Region Conservation Authority has been monitoring this population sporadically since 2015 and has witnessed significant mortality. So the salamanders likely overwinter in these green areas that I have on the map here. And the red road right in the middle is a very busy 2-lane road that connects to a 400 series highway. So it sees on average 19,000 cars per day. And then this blue road down at the bottom is a quiet residential road, 15 houses on it. It's a dead end. It only sees about 200 cars a day. So the issue, aside from the existing mortality, is that in the next couple of years, the red road will be expanding to a 4-lane road, which not only increases the amount of pavement that the salamanders have to travel across, but it also increases the traffic volume, both of which put salamanders at risk for mortality. And so the region wanted to know where the highest density of salamanders are crossing, or the road crossing hotspots, so that they can install eco passages under the road when they expand. As part of the current mitigation to protect these Jefferson salamanders, there is a soft road closure where they close the road to through traffic, but they let the residences drive through. However, in my experiences, the humans of Southern Ontario do not respect a large orange cone, sometimes driving through the cone, driving with it under their car for 800 meters or so. So we are not sure how effective this road closure actually is at reducing traffic volume or mortality. And so these road closures happen twice a year and occur for 2 to 3 weeks in the spring and fall, and they're only closed on rainy nights. So I have 2 objectives in this study. The first is to estimate road crossing hotspots, and the second is to determine if road closures are effective at reducing traffic. So in order to capture salamander movement, I surveyed 74 road surveys in the spring of 2022 and 2023, and 15 times in the fall of 2022. So starting 30 minutes after sunset, I surveyed Transect 1, which is that longer, busier road. I would then survey Transect 2, the shorter and quieter road, and then Transect 1 again, just to capture a larger range of time each night. Anytime we found a vertebrate on the road, we'd stop and let cars go past and then go and inspect. We ID'd it to species if possible and obtained a UTM location. If we found a Jefferson salamander, we took the snout vent length, total length, the sex, and life stage. And if it was dead, we removed it from the road to avoid recounting. If it was living, we would take it across the road in the direction that it was heading. In order to estimate hotspots, I used kernel density estimation in ArcGIS. So here are the results of the kernel density estimation from all 3 seasons. So we have adults on the left and juveniles on the right. And you can see that the adult hotspots are more concentrated than that of juveniles, denoted by the reds and oranges on the left-hand side. So 43% of adults are crossing within that 85-meter area. Juveniles, on the other hand, it's more diffuse. There's less concentration than that of the adults. And you also might note that where the juveniles are crossing is actually further away from the breeding pond than the adults. In order to look at the effectiveness of the road closure, we wanted to see how much traffic was getting through on the night when the road was closed. So we had traffic counters installed on Transect 1 and Transect 2 for 8 weeks during the spring of 2023. Once I got the traffic volumes, I wanted to see how traffic volume affected the probability of mortality. So we used an equation by Hels and Buchwald, where the probability of one individual being killed is equal to 1 minus e to the traffic intensity times the kill width. And the kill width is 2 times the tire width plus 2 times the salamander snout bend length divided by the velocity. And I decided to use snout bend length instead of total length because if a salamander were to get hit just on its tail, it would not likely die because they're able to regenerate. So that's why I used the snout bend length. And the last variable of the equation was velocity. So we had to run velocity trials to see how fast a salamander moved across the road. Unfortunately, my advisor didn't think I could use done et al. Studies. I'm not sure if any of you are familiar with it, but they put Jeffersons on tiny treadmills to test their endurance and their speed, but that wasn't realistic on a road. So we went out during road surveys, and I had volunteers stop anytime we found a Jefferson on the road. And whenever we'd find one, the volunteers would turn on the red headlights to not just disturb the salamander. And then they recorded the time and where the salamander originally started. So the trials were run for 10 minutes, but they ended early if the salamander finished crossing the road or if an oncoming vehicle was coming. The volunteers then measured the total distance the salamander moved as well as the morphometrics. And we were able to record 12 individual speeds. As you may have guessed, salamanders are not the fastest organisms. They move on average 0.35 meters per minute. Now, the speed also takes into account the 6 salamanders that would just sit on the road and not move for 10 minutes. But as you can see, some of them move quite quickly, almost 1 to 2 meters per minute. And at that point, they're basically just swimming across the road, which is pretty incredible to see. I did have my field tech sit and wait to see how long it took a salamander to travel across the road, the ones that just didn't move for the entire 10 minutes. And it took one individual 63 minutes just to finish crossing the road. So here are the results of the mortality model. And due to a technical problem with the traffic counter, we were unable to get an accurate number of cars that were driving through the road on a road closure. So instead, I decided to model when the road was open versus different levels of effectiveness of the road closure. So the number of cars per night from 9 to 5 are at the top there. And you can see on Transect 1, which is the furthest one to the left, with 2,100 cars passing through night when the road was open, the probability of death is 100%. If the road closure was 75% effective, the probability is still pretty high, 97%. If the road closure is 90% effective, so only 10% of cars were able to go through, only 76%, 76% probability of mortality. And then if the road closure was 99% effective, the probability of mortality dropped to 13%. So the blue bar at the far right is from Transect 2, the road that only had 240 cars per day, 16 cars per night, still had a 10% chance of mortality. So the probability of a salamander surviving when the road is open is basically 0. And only until we see a 99% effectiveness for the road closure happen, do we see a large drop in the probability of mortality. So adult Jefferson salamander road crossings were more concentrated and closer to the breeding pond than the juveniles. In past studies, most juveniles don't disperse more than 50 meters from their natal ponds, but at my study site, they were traveling up to 200 meters. I recommend installing eco passages every 50 meters along that road, just for ease of crossing for those smaller juveniles. And also eco passages should be monitored after installation. And finally, the probability of mortality for an individual salamander crossing that busy road was 100%. So salamanders do make frequent stops on the road, which makes them more susceptible to mortality. And against all odds, the population was able to persist even though, they are able to persist, which indicates that part of the population is probably living on the side of the road at the breeding pond. Road closures seem to be ineffective at eliminating through traffic, and more detailed reports are hopefully to come. In future work that I'd like to do, I'd like to model mortality using traffic volumes from other types of road across Southern Ontario, like paved county roads or dirt county roads and tertiary highways, etc. Just want to thank all of my funders and collaborators and open it up to any questions.
Hi everyone, I am going to share with you my deep dive into New Brunswick's common forestry practice and its effect on salamander populations and their habitat. As Leslie said, my name is Sara Leslie, and my project was supervised by Dr. Riley from the Riley Integrative Ecology Lab and Dr. Chris Edge from Natural Resources Canada. So I'll introduce you to my study species, which is the Plethodon cinereus, or I'll be referring to it by the Eastern redback salamander. So redback salamanders are excellent study species because they are vastly abundant in New Brunswick, being the most abundant salamander species in New Brunswick. They are excellent indicators of biodiversity and ecosystem integrity because they are sensitive to environmental change. So Eastern redback salamanders, unlike us, they don't have any lungs. So this means that they get their oxygen by breathing through their skin. And this cool feature, it does come with some limitations because this means to avoid drying out, they need to live in cool and moist environments. So typically, redback salamanders like to live in old growth forests because they have a high percentage of canopy cover. And this just promotes the cool and moist environments that they like to live in under decaying logs. And forests that can support a healthy population of salamanders is indicative of biodiversity and ecosystem integrity because these salamanders are a food source for vertebrates and they balance food webs, and they also regulate the capture of carbon by predating on invertebrates or capture of carbon from leaf litter decomposition by predating on invertebrates. And a fun fact is that these salamanders in healthy northeastern forests are estimated to be about 3 million eastern redback salamanders within a 1 kilometer square, which is a lot of biomass. And so I hope that you understand how these salamanders can contribute to the ecosystem by contributing to the nutrient recycling and trophic energy transfer in forested ecosystems. But locally, in New Brunswick, a main concern to salamander population is natural habitat loss, and specifically this is through forestry practices. And you can see on the image on the right is a control forest of an Acadian forest, which has a high canopy cover, and this is expected to have a high density of these eastern redback salamanders. Whereas in the image on the left is a clear-cut plot. And as you can see, there's drastic difference in canopy cover, which is expected to increase soil temperatures and lower soil moistures. And if you remember, this is the exact opposite of what salamanders like to live in their habitat. Along with clear cutting, and especially in New Brunswick, herbicides are also often applied when you are clear cutting while the forest is being replanted. And glyphosate-based herbicides are non-selective, so that means they will reduce or inhibit the growth of all vegetation. However, they don't inhibit the growth of coniferous forests or trees. So this allows the coniferous trees to grow a lot faster and dominate the forest. However, when you reduce the herbaceous layer, this is expected to change the environmental conditions in salamander habitats. And you can visually see this happening in the image on the right, where you have a clear-cut forest that did not receive herbicide application. And the image on the left is a clear-cut forest that did receive a herbicide application. And you can see that the untreated clear-cut plot has much less canopy cover at the same point of forest regeneration than there is in the treated clear-cut plot. So overall, the purpose of my research was to determine whether applying a glyphosate-based herbicide has greater negative effects on salamander populations after clear cutting than it does when a herbicide is not applied. So to explore my question, I focused on 3 objectives. My first objective was looking at salamander density, and this involved comparing redback abundance among forest treatments. Objective 2 focused on the indication of threats, and this involved comparing redback body condition among forest treatments. And objective 3 focused on reproductive activity, and this involved comparing redback juvenile proportion among forest treatments. So I predicted that if removing the herbaceous layer via herbicide enhances or extends the effect of canopy removal on redback populations, then salamander abundance will be higher, salamander body condition will be greater, and the proportion of salamander juveniles will be higher in clear-cut plots that are not treated with a herbicide. So I made these predictions with the understanding that if you remove the herbaceous layer when you clear cut, you're removing an extra layer between the hot sun and salamander habitat, and this may enhance or extend the negative effects of clear cutting on salamander habitat, at least during the first early part of forest regeneration. So my study design involves standardized concentric surveys where myself and a lab assistant walked 30 meters, 20 meters, 10 meters, and 0 meters from the center of a survey. And we looked 5 meters off of each side of these lines. So we looked for salamanders within the blue area of this diagram. And when we found a salamander, I visually assessed it for health by documenting any cuts or lesions. And I also looked for tail autotomy, such as missing or regenerating tails. And I also took morphological measurements like mass and length, and I recorded sex and species. Overall, I completed 30 surveys, so between 3 groups, and these groups included untouched forest plots that I used as my control, and clear-cut plots that did not receive a herbicide treatment, and clear-cut plots that did receive a herbicide treatment. And these treated plots were along a series gradient of 5 to 26 years post-harvest. And with the removal of the herbaceous layer, we believe to see the most notable effects on salamanders populations during the early part of this gradient, just because you're removing that herbaceous layer in these initial years. So before each survey, I also took environmental variables such as soil temperature, moisture, pH, and canopy cover. And this was just to test my initial assumptions of how the treatment of herbicides and clear cutting would affect a forest. For my results, I first looked at canopy cover, and unsurprisingly, canopy cover was greatest in the untouched forest compared to the clear cut forest. Notably, the canopy cover was significantly reduced in the untreated clear cuts compared to the treated clear cut forests. And this is also to remind you between the first 5 to 26 years, which is very interesting to see that within this short time frame, there is such a significant difference between the untreated clear cuts and the treated clear cuts. In this figure, you can further see that difference where within the around 25 years post-harvest, the herbicide plots have increased their canopy cover much more significantly than the untreated clear cut plots in blue. And similar to canopy cover, we again found the most number of salamanders in the control plots, and also the least number of salamanders in the untouched clear cut or the untreated clear cut plots, so no herbicides applied. And this is likely associated with the reduced closure of the canopy in the untreated clear cut plots, where in the treated clear cuts, you have your canopy cover recovering much faster. And this is allowing and promoting the salamander habitats to be more suitable within those later years of forest regeneration. And again, we have the number of salamanders on the y-axis and the years since clear cut on the x-axis. And you can see that the untreated clear cuts in blue, the abundance of salamanders stays relatively the same within years since harvest, but then the abundance of salamanders gradually increases in the treated clear cut forests. However, I didn't detect a significant interaction with this plot. And with body condition, I found the lowest body condition in the control plots, and the body condition was significantly different between my control and both of the clear-cut forest treatments. And this is likely or could be due to higher intraspecies competition in the control plots, where in the clear-cut plots, you have a lot less salamanders, which could make the resource availability much higher in the clear-cut plots where they have less competition with other salamander species. However, I do think there's more driving factors to this question. And lastly, we have no significant difference in juvenile proportion between or among any of my forest treatments. So to sum up, what did I find in terms of difference between my clear-cut forests? I found that there were more salamanders in my treated clear-cuts where the herbicide was applied compared to my untreated. However, I did not find a difference in body condition or juvenile proportion among my forest treatments. And overall, my research did not support my initial hypothesis that the removal of the herbaceous layer would enhance or extend the negative effects of salamander, of clear cutting on salamander population. Instead, my results suggest that herbicide treatment improves salamander environmental conditions for their habitat compared to the untreated clear cuts, and within these early stages of forest regeneration. So I see value in continuing this research to investigate how these common forestry practices affect salamander food availability and downwoody debris, as these are very key components in salamander habitat quality. And I'm also interested to see if there's similar trends observed in later stages of forest regeneration, whether you see a different trend or if you find the similar trends that I'm seeing now. So I hope that if you've learned anything from my presentation, it's that salamanders are not just cool creatures that you can find under logs when you look for them in the forest, but that they can tell you so much about a habitat's health, and they can help provide information about common forestry practices, especially in New Brunswick, where I did this research. So I want to thank everyone who helped me with this project, especially my supervisors, Dr. Riley and Dr. Chris Edge, and especially my funders who helped me do this. Thank you.